ever think about uh, building proteins like Legos? Kind of crazy to think about, right? Like swap out pieces to make them work better.
Yeah.
Well,
It's a cool thought.
that's what we're diving into today.
It is.
This 2024 Cell research paper, it unveils ProMEP.
Right.
It's this AI tool and
Right.
basically turning this Lego dream into reality.
Yeah, it's not just cool, though. It's like
Yeah.
potentially a game changer for fields like biomedicine and synthetic biology, you
It's
know?
totally. I mean, that's where we're taking a deep dive into the research today.
Right.
Picking out the most fascinating parts for you guys listening.
Yeah.
But before we even get to the AI wizardry of ProMEP,
Yeah.
let's just start with the basics. Proteins and those tiny changes, mutations that can just make all the difference.
The paper really highlights how even a single amino acid change in a protein sequence, it can have this ripple effect on its function.
It's kind of mind-boggling when you think about it, right?
Yep.
Like how these tiny little tweaks can be behind everything from how well our bodies work to how viruses evolve.
Absolutely.
It's crazy.
Yeah, and that brings us to, I guess, the challenge that ProMEP is tackling head on.
Yeah.
The vastness, the sheer vastness of possible protein sequences.
Right.
It's like trying to find a needle in a haystack the size of a planet.
Right.
You can't experimentally test every single mutation.
There's just
It's just not feasible.
no way.
Yeah.
So enter ProMEP
Sorry.
stage left
Stage left.
with its multimodal deep representation learning.
Right.
I know that sounds like something out of a sci-fi movie, but bear with me.
Well, think of it this way, right?
Okay.
ProMEP is like a super smart chef
Okay.
who not only knows like every ingredient in the cookbook,
Mm-hmm.
but also understands how those ingredients all interact with each other
Okay.
to create, a delicious dish.
Right.
In this case, the ingredients, those are the amino acids in a protein. And the dish, well, that's the protein's 3D structure and function.
Oh, so it's not just looking at like the individual pieces.
Exactly.
It's figuring out how the whole puzzle comes together
The whole puzzle.
and how they fit in 3D space to actually do something.
Exactly. And that's what allows it to predict the effects of mutations with such remarkable accuracy. It's like being able to predict how changing one spice in a recipe
Yeah.
is going to affect the final taste and texture of the meal.
Okay. Yeah, that makes way more sense.
Yeah.
So it gets even more impressive, though.
Yeah.
This ProMEP can also do something called zero-shot prediction.
Right. Which means it can make predictions about proteins it's never even encountered before.
Oh, wow.
It's like meeting someone new
Okay.
and being able to like accurately guess their personality
Mm.
based on your knowledge of other people.
Okay.
Yeah.
So it's basically using what it's learned from other proteins
Exactly.
to make educated guesses
Yeah, educated guess.
about new ones.
All
That's
right.
pretty slick.
Oh, it really is. And this zero-shot learning, it's a game changer, especially when you're dealing with proteins that maybe haven't been studied that extensively.
Right.
Or they don't have, many known relatives.
How does ProMEP even do that? It's like it has like protein intuition.
Well, that intuition,
Yeah.
it comes from its attention mechanism.
Okay.
It analyzes how amino acids interact with each other, both sequentially, like the order of words in a sentence,
Right.
and spatially,
Yeah.
like how those words fit together to kind of like create the sentence's meaning.
So it's understanding the protein's like grammar as well as its vocabulary.
Precisely. And this ability to analyze both sequence and structure,
Oh.
well, that's what makes ProMEP so much faster
Right.
and more accurate
Than previous methods.
than previous methods like alphamescence.
Oh, okay. So we've got this super-powered AI
Yeah.
that can predict mutation effects with incredible speed and accuracy.
Right.
But how is it actually being used in the real world?
Right.
The paper highlights two fascinating applications. The first one focuses on TNPB,
Okay.
which is an enzyme that plays a crucial role in gene editing.
Now, gene editing is already a pretty hot topic,
Yeah.
but TNPB is especially interesting because it's an RNA-guided nuclease,
Okay.
which means it can be programmed to target specific DNA sequences. So it's like a tiny molecular surgeon that can make these super precise cuts in DNA.
That sounds incredibly powerful.
It is powerful.
Yeah.
And with ProMEP's help,
Mm-hmm.
researchers were able to engineer like a version of TNPB
Okay.
that's even more efficient.
Okay.
They created a quintuple mutant,
Okay.
which basically means they changed 5 amino acids.
5 changes. And what? That just like made it a super powered gene editor.
Pretty much, yeah.
Wow.
This engineered TNPB showed significantly enhanced activity,
Okay.
meaning it was much better at cutting DNA at its target site.
Right.
And this led to the development of, like, these improved base editors,
Huh.
which are basically like super precise gene editing tools.
So we're not just talking about understanding mutations now.
Right.
We're talking about using that knowledge to engineer better tools for gene editing.
Exactly.
That's crazy.
It's a whole new level.
Yeah.
And the second application that's highlighted in the paper focuses on another enzyme called TADA,
Okay.
which is also involved in gene editing.
So they use ProM to give TADA an upgrade too.
Yes, but in a slightly different way.
Okay.
This time they engineered a TADA mutant with reduced bystander and off-target effects.
Hold on, back up a second. What are bystander and off-target effects?
Okay, think of it like this.
Okay.
When you're editing a gene,
Yeah.
you want to make sure you're only changing, like, the specific part of the DNA that you're actually targeting.
Mm-hmm.
But sometimes
Yeah.
the gene editing tools, they can kind of make accidental cuts in other parts of the genome.
Oh, so like a surgeon accidentally nicking something they weren't supposed to during an operation?
That's a good analogy. And these unintended cuts are what we call off-target effects.
Right.
And they can lead to, like, unwanted mutations,
Oh, no.
potentially cause problems, yeah.
So by reducing these off-target effects,
Right.
the engineered TADAs is basically like a safer and more precise gene editor?
Exactly. And that's a huge deal because it
Right.
means we can potentially use gene editing in a much more controlled and reliable way.
Right. Okay, that makes sense.
Yeah.
So we've got ProMEP helping to create both more efficient and more precise gene editing tools.
Right.
That's pretty amazing.
It really is.
Yeah.
And these are just two examples of how ProMEP is already being applied.
Mm-hmm.
The paper emphasizes that its potential applications are much broader.
Like what else could we use ProM for?
Well, for starters, it could help us design, like, entirely new proteins
Oh.
with, like, enhanced functions,
Okay.
like enzymes that break down pollutants, for instance,
Right.
or antibodies that target specific diseases more effectively.
So we could basically start creating custom-made proteins for all sorts of purposes.
Exactly.
That's incredible.
And that opens up a world of possibilities in fields like, medicine, agriculture, environmental science.
Okay, my mind is officially blown.
Yeah.
But I have to ask,
Yeah.
if ProMEP can predict how mutations affect proteins,
Right.
could it also be used to predict, like, how viruses might evolve?
That's a great question.
Yeah.
And it's definitely something researchers are exploring.
Mhm.
Imagine being able to, like, predict which mutations in a virus
Yeah.
might make it, say, more contagious.
Right.
Or more resistant to treatments.
That would be a game changer for public health.
Absolutely.
And, like, pandemic preparedness.
Yeah.
I mean, we could potentially develop vaccines and treatments so much faster.
Much faster.
Yeah.
Exactly. And that's just one example of the potential impact
Right.
that ProMEP could have on our understanding and management of infectious diseases.
This is all incredibly exciting, but it also makes me wonder about, like,
Yeah.
the limitations of ProMEP.
Sure.
Is it perfect? Are there things it can't do?
Well, no tool is perfect.
Right.
ProM's predictions are based on the data it's been trained on,
Mm-hmm.
which means there's always a chance it might encounter, a protein or a mutation
Right.
that it just hasn't seen before and make an incorrect prediction.
So it's still learning and evolving, kind of like the proteins it's analyzing.
That's a great way to put it.
Yeah.
And that's why it's so important to remember that, ProMEP is a tool.
Right.
It's a powerful one,
Yeah.
but it's still just a tool.
Right.
It's up to, scientists and researchers to use it responsibly
Mm-hmm.
and to interpret its predictions carefully.
Makes sense.
Yeah.
So we've covered a lot of ground here.
We have.
We've talked about the power of ProM to predict the effects of mutations,
Right.
its role in engineering better gene editing tools,
Right.
and even its potential for predicting viral evolution.
Yeah.
What does all this mean for the average person, like someone who might not be a scientist
Right.
or a researcher?
Well, I think the key takeaway here is that ProMEP is like a prime example
Mm-hmm.
of how artificial intelligence is revolutionizing our understanding of biology and our ability to manipulate it.
And that's something that's going to affect all of us, whether we realize it or not.
Absolutely.
Yeah.
The advancements in protein engineering that ProMEP is driving, they have the potential to lead to new treatments for diseases,
Right.
more sustainable ways to produce food and energy,
Wow.
a deeper understanding of the very building blocks of life.
It's like we're on the verge of being able to not just read the language of life,
Yeah.
but actually like write it and edit it
Right.
Maybe even compose entirely new stories in that language.
That's a great analogy.
Yeah.
And the fact that we can now use AI to do this so efficiently and accurately
Right.
is both exciting and a bit daunting,
Taunting for sure, yeah.
don't you think?
It's like we're opening up this whole new world of possibilities,
Right.
but we also need to be really careful about how we navigate it.
Absolutely. And that's why I think it's so important for everyone, not just, scientists,
Right.
to be aware of these advancements
Hmm.
and to engage in conversations about the ethical implications.
So this isn't just a deep dive for the science nerds out there. It's
Yeah.
something that everyone should be paying attention to.
Absolutely.
Yeah.
Whether you're interested in, healthcare, food security, environmental protection,
Hmm.
or just like the future of humanity in general,
Right.
protein engineering is going to play a huge role
Yeah.
in shaping that future.
Okay, so for all of you listening out there, here's the bottom line. ProMP is this powerful tool
Hmm.
that's already making waves in the world of protein engineering,
It is.
and this is just the beginning.
It's a field that's moving incredibly fast.
Yeah.
And the research that, we've been discussing today is really just a glimpse of what's to come.
We've seen how ProMEP can predict the effects of mutations with incredible accuracy. How it's being used to create more efficient and precise gene editing tools,
Right.
and even how it might help us predict the evolution of viruses.
Yeah. It's pretty remarkable.
It's mind-blowing to even think about all the possibilities.
It is.
But it also, kind of underscores the need for careful consideration
Yeah.
of the ethical implications.
For sure.
We need to make sure these powerful tools are used responsibly
Right.
and for the benefit of all humanity.
Well said. I think that's a perfect note to end on.
Yeah.
This deep dive has hopefully given you, listening out there, a better understanding of the incredible advancements
Yeah.
that are happening in this world of protein engineering.
And we encourage you to keep learning, keep asking questions, and keep engaging in these conversations that'll ultimately shape the future of this revolutionary field.
Couldn't agree more.
Because understanding these advancements isn't just about keeping up with science. It's about understanding the future that's being built, one protein at a time.
One protein at a time.